current challenges associated with making traditional IgG monoclonal antibodies really stems around the high level of complexity of these products, and bispecific monoclonal antibodies in the pipeline are even more complicated. So for the foreseeable future, these are likely to remain high-cost products and only available to those that can afford them. But VHHs are amazing molecules. They are quite robust and stable. They have high solubility. They're exceptionally well suited for expression in Baker's yeast systems. They have a number of properties which lend themselves especially well to this eukaryotic microbe. So Baker's yeast has a long, safe history of manufacture, over 35 years, for example, with products like insulin. And it has the benefits of being a microbial system. So it has some of the advantages of E. coli, it grows quickly. But it also has some of the advantages of mammalian systems, like being able to perform eukaryotic post-translational modifications. So the marriage of these two systems, VHH and Baker's yeast, is particularly attractive.